Hello everyone, welcome to ITLS Academy. Our lecture is Genital Culture and Sensitivity Test. First of all, we will see aim of the test or why the genital culture test is performed. So the first point is the aim is to isolate and identify potentially aerobic pathogenic organisms including Gardnerella vaginalis and group B Streptococcus. Establish the diagnosis of gonorrhea, medical legal cases. These are aims of the test. A genital culture allows the organisms present in the vagina to grow to levels enabling identification. The vagina is not sterile but contains a mixture of aerobic, anaerobic, coliform and skin bacteria as well as a few fungi. Normally, these are in balance with no particular predominant organisms. Next point is significant growth of any one organism usually indicates a clinical or subclinical infection. Here, some common types of overgrowth include yeast like Candida albicans, Streptococcus, Gardnerella, E. coli, and Proteus. Types of specimen. Types of specimen include swab of vagina, cervix, discharge, aspirated endocervical, endometrial, prostatic fluid or urethral discharge. Use swab to inoculate germs for transport to the laboratory and recovery of Neisseria gonorrhoeae. Swab should also be sent in transport device. This is all about types of specimen and their transport. Collection of specimens. this diagram is showing the collection process, how this particular person is collecting a specimen. This diagram is showing vagina, uterus, spatula. This is spatula. Someone is holding a spatula, and from the help of spatula it is collecting the fluid or discharge from this particular area as specimen. This is speculum. This area shown over here is speculum, and this is cervix, and the sample is taken from the cervix. This fluid is taken by the help of spatula. Two points regarding collection of specimens are given. Let's see them one by one. First point is the specimen commonly collected for the diagnosis of vaginitis, vaginosis, or uterine sepsis is high vaginal swab. So when there is any kind of disease like vaginitis, vaginosis, then specimen collection, then specimen collected is high vaginal swab. Next point is the swab is inserted into upper part of the vagina and rotated there before withdrawing it. So this is collection of specimens from males. So first we will see how the specimen is collected from females. Here you can see in the case of females it can be collected from cervix or it can be collected from vagina, while in the case of males it is collected only from urethra. So we are going to study females, how the specimen is collected from specimen. Firstly, we will see specimen collection from cervix. First point is remove mucus and secretions from the cervix with a swab and discard. Next point is with a new sterile swab formally sample the endocervical canal. Third, place in swab culturate. This is the process of collection from the cervix. Let's see how the sample is collected from vagina. First point is wipe away excessive amount of secretions or discharge. Next point is obtain secretions from mucosal membrane of the vaginal wall with a sterile swab. Third point is if smear is also requested, collect a second swab. And the fourth point is place swab in culturate. Next we are going to see In the case of males, how the sample is collected? So in the case of males, it is collected from urethra. First point is insert a urethro-genital swab 2 to 4 cm into the urethral lumen and rotate swab. Second point is leave in place for at least 2 seconds to facilitate absorption. Third point is place swab in culturate. Criteria of specimen rejection, or the conditions in which conditions a specimen can be rejected. Inappropriate specimen transport device, mislabeled specimen, unlabeled specimen, specimen received after prolonged delay, usually more than 72 hours. Here you can see in most of our previous tests, we have seen that this delay period is more than 2 hours, while in the case of genital culture test, it is more than 72 hours. Next criteria of specimen rejection is specimen received in expired transport media or expired transport media containers. Some other criteria of specimen rejections are improperly labeled specimen, specimens with prolonged transport time or transit time, specimens not submitted in appropriate transport containers, specimens having insufficient volume, specimens having external contamination. If an unacceptable specimen is received, the physician or nursing station will be notified and another specimen will be requested before the specimen is discarded. Next topic is transport or storage of specimen. On-site collection. In the on-site collection, transport to the microbiology laboratory immediately. Do not refrigerate. Refrigeration may prevent the recovery of Neisseria gonorrhoeae. In the case of off-site collections, do not refrigerate, store at room temperature. Specimens must be promptly transported to the laboratory with the next available courier, not to exceed 24 hours from the time of collection. However, a delayed transport causes a delay of test results. Here, some pathogens and their commensal floras are given. In the case of genital tract infection, these are pathogenic bacteria and these are their commensal flora. Pathogenic bacteria are Neisseria gonorrhoeae. Commensal bacteria is coagulase negative staphylococci. Group B streptococci and commensal bacteria is Corynebacterium species. Next pathogenic bacteria is Gardnerella vaginalis and commensal bacteria is E. coli and other coliforms. Pathogenic bacteria is Enterococcus species while commensal bacteria is many species of anaerobic. Pathogenic bacteria include certain anaerobes including Actinomyces species, Haemophilus duckreyi, Trypanoma pallidum, Mycoplasma species and Enterobacteriaceae, and last but not least Chlamydia trachomatis. These were some pathogenic bacteria. Let's see some pathogenic fungi, that is Candida albicans. In case of parasites, it is Trichomonas vaginalis. In case of viruses, it is herpes simplex virus and human papilloma virus. These are some pathogens and their commensal flora. Pre-specimen processing. First one is specimen collection. In case of females, do not use lubricant or speculum. Cervical mucus should be removed first before inserting swab into endocerv side to side, allowing several seconds for absorption of organism by the swab. Return swab to the transport tube and label. In the case of specimen collection from males, using small wire swab, gently scrape the anterior urethral mucosa or use a swab to collect a specimen of urethral discharge. Quantity of specimen, sufficient amount on swab. This is quantity. Time relapse before processing the sample is 30 minutes. Storage condition is maintain specimen swab at room temperature. Do not refrigerate. Specimen processing. The media includes blood agar, that is 2 plates, chocolate agar, McConkey agar media and sub agar. Let's see what are the culturing procedures. Culturing procedure is streak 2 blood agar plates, 1 chocolate agar plate, McConkey and sub agar plate. Do wet mount, examine for clue cell and trichomonas, and gram stain to check the predominant organisms. Here is culturing process is given. You can see this is genital specimen. Genital specimen is taken, gram staining is done, and after gram staining there is another process, that is wet mounting. Wet mounting is done, and then this specimen is given for culture. Culture include blood agar, chocolate agar, McConkey agar and sub agar. This is the culturing procedure. Here comes post-specimen processing. Post-specimen processing include interfering factors. Interfering factors include first one, patient on antibiotic therapy, and the second one is improper sample collection. Next is result reporting. Result reporting include number 1, report wet mount gram stain finding as an initial report. Next, report the isolated pathogen and its sensitivity pattern as a final report. Turnaround time. Number 1 is gram stain and wet mount results should be available 1 hour after specimen receipt. Next point is isolation of a possible pathogen can be expected after 2 to 3 days. Negative culture will be reported out 1 to 2 days after the receipt of the specimen. Here diagrams of genital cultures are given. You can see this is the sample collection procedure. This is urethral and nearby structures are given. This is seminal vesicle. This is rectum part of the body. This part is vas deferens. This is anus, that is opening of the rectum. This is bladder and this part is ureter, tube-like structure. This is prostate. This one part is urethra. You can see this is pipe-like structure is urethra and this is penis. To overcome some of the problems involved in obtaining adequate amount for the recovery of organisms in suspected cases of sexually transmitted disease, kits containing swabs, slides suitable for collecting such specimens are now available and could be used in physician offices. This was all about genital culture. Thank you for watching this session. You can follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn. For latest updates and videos, you can subscribe ITLS Academy on YouTube and you can also get it on Google Play Store. Thank you very much for watching this session. You can like, share and subscribe ITLS Academy. For any help or query, you can call us on 7080-833-450. You can also reach us on www.itlsacademy.com. Thank you very much again for watching this session.